morning session. Um, Thank you for sitting through this as you have your lunch. This is something different, as it was mentioned. Different, first of all, because I'm not a medical doctor. So I would like to ask anybody who's not a medical doctor and it works with data. Is anybody in here? That's no. Well. However, you all have worked with data. You set through a number of talks, and you've seen a lot of numbers, a lot of statements made based on data. So why should you care? You should care because clinical trials is the standard, the gold standard, based on which decisions are made in medicine. Decisions are informed by using clinical trials that use only a sample. That means that statements that are made come with a certain amount of uncertainty, and statistics provides that language through which the uncertainty is communicated. Whenever you see certainty coming from data, you should be very suspicious. When you see a estimate that doesn't come with a number that doesn't come with some quantification about uncertainty around it, again, you should be suspicious. When you see even close to certainty, again, you should be suspicious unless you see something maybe coming from the census. Properly accounting and communicating the uncertainty is important because you might get your moment of glory in which you pretend that you're very certain, and then very soon you realize that you were proven wrong and that uncertainty really came back to bite you. Why you should care about such a talk is that also, you've seen different body of evidence, and you've seen some of the talks in which people really tried to explain different results coming from different sources, and that's also its uncertainty in action, and having some way of getting these disparate sources of information and producing a conclusion or a statement that unify these sources is also very important. So that's why maybe you might want to sit through this. And the last but not least reason of why I think you might want to sit through is that one of the reasons of concern that people who work in clinical trials is that they under-enroll. They have problems in enrolling. If you read through many clinical trials, you'll see that that this is the reasons of why clinical trials fail. This is one of the reasons why clinical trials are prolonged, because patients are not found and cannot be enrolled. So having some way of being optimal is necessary for us to be able to run clinical trials, and it's also the ethical things to do. It's hard to stay in front of a patient and say, well, because I'm unable to really standardize or summarize all the information available, I'm going to rely maybe on a toss of a coin to assign you to a therapy. And it's ethical, it's the ethical thing to do to really take all the information, because once you know, or once you have a good, you don't know again, once you have some certainty of one treatment being the other, you should not randomize anymore, because the patient that's in front of you expects that you provide the best advice and the best medical advice, and you use the best treatment available, given the evidence. So the key word in my talk was the Bayesian. Oh, sorry. That's, I don't know how that title got in there. It's Bayesian approach, Bayesian framework in making decision with clinical trial. So why Bayesian, and why not just statistics, using statistics in making clinical trials? Because there are two main currents in statistics, and that's more like philosophical approaches, a way one can think about uncertainty, one can think about making statements based coming from the data. Majority at least majority of the stats that we've seen in the presentations were based on the dominant kind of way of thinking about statistics, and that's the frequentist approach. And why is that? Why were most of the studies are run and decisions are made using this philosophical way of thinking about statistics, is because there were a few very influential people that kind of imposed this approach, this way of thinking about statistics. Now, we all learn about thinking about null hypothesis, about alternative hypothesis. We're all looking about p-values, and we're all looking about confidence interval. These are the concepts within the frequentist approach, and this is the dominant approach in clinical trials. And sometimes we talk about probabilities, and very few of us think about where are these probabilities coming from. And just to give you the main source of probability, and that is how do the results of the particular study that you just run fare in a long run of studies that are run under exactly the same condition. And if you're trying to think literally about this, you would realize that this is an impossible construct. There is no way that you can run a same trial under many conditions, because unless you do it by the same, using the same investigators under the same exact conditions, and at the same time, it's very impossible to have this identical reproduction of the same study. So the probabilities that we're talking about are coming from this interpretation, and if you're including p-value, including confidence intervals, and if you're put in the position as a physician that you're saying, well, I'm going to recommend this treatment because that difference is significant, and that's really coming from the fact that you have a p-value that possibly is smaller than 0.05, depending on the situation. I would challenge you to try to explain that concept to a patient in front of you, and I think will be very hard. And the question that I would have is, how does that apply to me? Why should I trust this interpretation that might have to look at other trials that have not been run, or that might be impossible to run? So I'm not going to bore you with some of the concepts, but I'm going to present you and just give you some of the concepts in the alternative approach that to statistics, and that's the Bayesian approach. And to the reason we run this statistics was we try to learn something about a quantity of interest. Here being like the mean or a rate of a disease, in this approach, you're forced actually to think, what's the state of knowledge? And then, given that state of knowledge, you're asked to incorporate the evidence collected within your study and create a new state of knowledge. And that state of knowledge includes certainties and uncertainties. And the language to communicate and the language to talk about uncertainty in this approach are probabilities. Probabilities are used in everyday life in expressing uncertainty. You know, yesterday we were thinking, given the rain that we've seen and the temperature, maybe it's really likely we're going to have more rain today. And that was based maybe on our prior experience on the weather during May, and in this place, not someplace else, and during this year. If we're getting into the business of interpreting it in the long run, we would have a problem, because based on long run, we know that kind of it's warm, and now we keep having this cold weather, a lot of rain. So based on only this year rather than many years, we would, in our experience, based on yesterday and possibly peeking a little bit at the Google weather report, we might say maybe it's very likely we're going to have a rain. So we use probability we can't say it will be for sure, but probabilities is the language that people do express uncertainties and certainties at the same time. So in the Bayesian approach, that's the case. Because we don't know the values of these parameters, so we have uncertainty around what the true value is, we're going to have a probability distribution modeling what that value is prior to running the study. And of course, after we update this, then what the result will be a probability distribution that will express, again, certainties and uncertainties that were updated based on the evidence that we just observed. So I'm not going to go more, given the audience and or your interest. I'm just going to go through some examples of trials in which I've been part of, and some of this, some of the directly or indirectly. And I try to choose the examples so that might be relevant to your to the cardiology in general. So how do we go and express, judge, make decision based on what? Of course, would be based on probabilities. No, I'm sure. I hope you get somehow this in your habit, because the decisions will involve. We won't be able to say for sure this is better than this, but we can provide some type of, use the language of probabilities to express how certain we are and how uncertain we are. And because we're trying to evaluate some type of hypothesis of the research hypothesis, rather than providing a p-value whose interpretation again will involve this repetition or replication of the same trial, would have just the probability of having a positive result as the final the way of communicating the decisions that what we based our decision on. And so in that, it's similar that we're calling a p-value like some type of a probability, and the posterior probability of the research hypothesis being another. The interpretation, the philosophical thinking about this is very different. Again, p-value involved this replication, while this the probability of the research hypothesis only involves the observed data. So condition of what we've seen and what's here, what's actually hidden here, and condition on our previous state of knowledge before collecting this data, which is the prior. That's our conclusion. That's our probability that the research hypothesis is true. So what's the research hypothesis in a clinical trial? Usually, well, the proposed intervention is better than the standard intervention. And that's really the direct quantity that we're interested in. And this approach also allows incorporating in decision-making maybe some economical factors. And in that way, you put together some type of utility or cost function that might involve the cost of the proposed intervention, the size of the population that you're targeting this population, and that might give you a framework of making a more holistic decision rather than just p-value smaller than 0.05 or not. This decision, besides the probability of having a positive result, you also incorporate cost, again, size of the population, and so on. So these are some examples that I've been kind of directly or indirectly involved in. I'm not going to cover to go through all of them. I'm going just to sample a couple of them and then give some time for discussions and questions. I'm going to skip this one given the time. So yeah, this one I'm not going to skip. So this is the example of a trial that we designed for Biotronic, which is a company that's based in Switzerland, and was for the evaluation. Was for the evaluation and effectiveness of a Orsiro stent as compared to a Xience stent. And the interesting feature of these situation was that the company actually had two studies that they already run. One of them already finished, that was BioFlow 2, and one of them that was still under follow-up and recruiting the last patients at the time of the design of the ultimate approval study. So we had the option with a traditional approach. You might use these two studies to maybe inform the design of the approval study. However, the quality of these data was pretty high, and rather than ignoring these data, the proposed study suggests the study proposed to use the data from these studies, while of course, given this regulatory requirement, while controlling the type 1 error and type 2 error, meaning the chance of making a false negative or a false positive statement. The decision was, this was going to be a non-inferiority trial, and the decision was that when the probability of having a positive result, which is a non-inferiority hypothesis, given the observed data and prior data, which was BioFlow 2 and 4, exceeds a certain threshold, which was a priori agreed with the regulatory agencies, then the decision was, or the agreement was, to make the decision to declare non-inferiority. If the probability, after considering again all evidence, is smaller than this, the decision would have been that this will not continue. So the task was to integrate these while recognizing that potentially we will observe some results that will not exactly match the results of the proposed study. It's just blindly taking the data from these three studies, shuffling them, forgetting where the study coming from, would, first of all, would be the wrong thing to do. Second of all, would be the unacceptable thing to do from the regulatory perspective. And what the decision was to assume that the rate in the proposed study would possibly differ with a bias term between both groups, the interventional, which was the 0, and the science group. And the size of the bias came from information regarding the similarities of the studies, and the comfort with making this assumption came from the fact that we performed several, we took several measures, among which we had the same team re-adjudicate all of the events from the historical study and from the current study. And after some statistical evaluations, we came with some actually values that bound the magnitude of the bias and allowed us to estimate and integrate this data and allow for a robust approach that would not provide a high type 1 error and type 2 error in the case you were surprised. And this is some information about the size of the bias. And here you can see that when allowing for bias term, we got a nice bound on the type 1 error, and we had reasonably good power. While when ignoring a bias term, we had high power under all of the assumptions. However, under some assumptions, the type 1 error was really inflated. So while this approach had some loss in power, was more robust in the sense that when the historical study and proposed study are disparate in some way, then we'll still get a pretty good bound on the type 1 error. And just to, this study finished and actually ended up with a positive results. Because there was so much talk about TAVR, there were 2 studies run by Medtronic. We were involved in Baim Institute as validation group. They proposed a Bayesian design for both for the intermediate risk patients and for low risk patients, in which the Bayesian thinking was used in predicting data for unobserved information that at the interim looks. And what this approach allowed for bringing the proposed interventions to the regulatory in advance of study finishing, and that was so this used what was the advantage, first of all, of using the Bayesian approach. Bayesian thinking was that prediction of unobserved data was more realistic. Of course, you got more uncertainty. However, the prediction results were more reliable. And so this used a very simple imputation model, while the low risk used a more sophisticated prediction model, in which was recognized that the risk of events early in the follow-up is different than the risk of events in the mid follow-up, and different than the risk of events towards the end of follow-up. And just to give you a taste of how this how the results are communicated, of course, probabilities, and rather than having a p-value, you get that interval. You get the posterior probability of TAVR being better than SAVR is 0.999. So that's not 1, but it's pretty confident that the research hypothesis is true. And then you get estimates. You see, you should know the B here instead, to make sure that this is not interpreted as confidence interval. This is Bayesian confidence interval. But this was usually, these are called credible intervals. Just to make the point that these are not regular confidence interval. And by the way, if you do your interpretation, I'm sure that you're very likely that you're doing the right interpretation. But if I ask you to interpret the confidence interval, likely that you might get it a little bit wrong. While this is just the probability that the true value, true event rate for TAVR is between 3.3 and 8%. So just to end before it's too late, I just don't want to be late. There are many other examples of this application of the Bayesian thinking, and what the advantage. It's using the probabilities and around expressing uncertainty. I think that's the most distinctive feature of this approach. Conclusions are easier interpretable, easier to communicate. For example, would be that there is a X% probability that treatment A is better than treatment B. This is a little bit, this should have been. Bayesian approach can result in more flexible methods for integrating available information in decision making. There are more resources needed. There is more thought to be put into design, and it's more patience. And for this to succeed, you need a savvy PI, a person who's ready to sit up and think through the available information, think to the compatibility of the available information with the existing study, and of course you need sponsor buy-in. Any questions?
Professor Dorosh, thank you very much for your really excellent presentation. Don't ask me to repeat anything, please. I'm just a cardiac surgeon. And now the paper is open for the discussion.
Maybe I just one maybe two simple questions, but at least for you. But okay, we have seen what is the what are the positives from the Bayesian approach. But in your opinion, what would be the main disadvantage of Bayesian approach?
Yeah, that's a good point. Well, it's resistance from the existing community, I think, and the necessity of learning. It's a learning curve for the people who might be interested in applying this methodology, and I listed as a positive the necessity or the possibility of incorporating prior information, but this is often listed as a negative because people say, well, now I have to summarize all the prior knowledge about the quantity of interest in a form of a probability, and that's not obvious all the time. And because, as I mentioned in my I think second slide or third slide, the Bayesian machinery will take this prior information and will combine with the existing data to give you an updated posterior information. So there are three components here. So the prior information is one of them. So that means it has an influence of the end result. So if you do the wrong thing, if you're not doing the right thing in summarizing the evidence, you might get the wrong result because all these three count. And so it requires a lot of thought. When you ask many physicians about, give me the probability of this, and I say, what's probability? So there is a necessity to translate between the very rigorous mathematical expression of what a probability is and the way a physician thinks about probabilities to faithfully translate what a physician thinks into a mathematical object, which is a probability distribution. So so those are so it could, you have that flexibility, that you have the responsibility of using it right.
So if we translate that into the clinical practice, it should be it's operator dependent. So it's very dependent on who is doing the analysis.
Exactly. Yes.
Yeah. Okay.
Okay. Thank you very much. For sorry, please, Joseph.
Yes. Yes. Actually, I never do meta-analysis in the traditional approach. I always use the Bayesian approach. Why? Because and I had an example about it, but I skipped over. It's about DAP study, in which we conducted a meta-analysis to explain the unexpected rise in non-cardiac mortality with longer-term use of DAPT. Again, non-cardiac mortality with longer-term use of DAPT, and this is being observed in DAPT. But so we had to look at all the body of evidence up to DAPT, including DAPT, and that was done using a meta-analysis to try to explain the unexpected. And here you have to bring to the table all the uncertainty around the heterogeneity in the study results, uncertainty about knowledge of this uncertainty, which is a very important, very influential quantity that is mishandled by the classical approach. Now, we always talk about tau squared, about I squared. Now, we run a homogeneity test and to feel comfortable. But at the end of the day, taking the classical approach basically has a guess about heterogeneity, and variance very problematic. Sometimes there's no uncertainty. Just the latest is the meta-analysis around the paclitaxel DES and DCP, which is you have 27 companies in 12 countries, or sorry, 12 countries, 28 studies run over time, and if I ask you about before looking at the data, are these results homogeneous? I would say no, because many countries, many companies, many groups. However, the traditional meta-analysis comes up with a response. There's no difference between these studies. With the Bayesian approach, you actually get an estimate, and then basically some average across all these homogeneity results that are obtained through of course, a model. So that in terms of meta-analysis, I never run, if you look at any papers I involved with, we always run Bayesian meta-analysis. And the community is pretty accepting of that. You're not getting, well, no, I don't want to look at that.
Thank you very much. For the sake of time, we have to go on. So the following lecture is going to be given by Dr. Smiris from Germany, aortic root surgery, and then Dr.